Hi everyone, my name is Juan Ángel Vaquerizo and I'm part of the NIC Spain, and I'm going to share with you some hands-on activities about astrobiology in this talk titled Looking for Life in the Universe. First, I'm going to talk with you about some general ideas about astrobiology, if you don't know what it's about, and then I'm going to share with you some hands-on activity that we are doing with the students in this kind of topics, okay? So first of all, astrobiology is the study of life in the universe. It means that we are trying to understand about the phenomena of the astrobiology, the phenomena of life in this universe, and that make us to doing questions about the presence of life, not only in our planet Earth, but in other planets around the universe. So we need to start with some kind of hypothesis, a scientific hypothesis regarding astrobiology, and that's it, that life as a phenomenon is a consequence of the chemical evolution of the universe. So we are studying life from a chemical point of view. It means that for us, life is the result of the evolution of matter in the universe, not in all the universe, but in some local places as planets and moons. In this big picture, we try to explain about the evolution, the timeline of the universe, starting in the upper left part of the picture with the Big Bang, and then finishing in this only planet with life by now, planet Earth. So we try to understand how is it possible in the universe to create life in some specific environments. And this is done in a multidisciplinary approach. This is not only regarding biology, but also chemistry, of course, but also geology, physics, astrophysics, mathematics, engineering. So we need every single scientist in the planet to try to understand what is life first, and then if this phenomena, this life, it's possible outside our planet. In a very general point of view, we try to understand in a very integrated way about biological, planetary, and cosmic phenomena. It means that our level of focusing or zooming is from local, biological in our planet, then planetary, all the planets, all the environments in the universe, and a cosmic point of view. It means that we are trying to understand in the whole universe where the conditions can make possible for life to exist. We need to search not only for life, but also for habitable environments. It means that we need to know about the environments where life could exist, and we need to develop certain techniques to analyze this environment and to try to understand if life could be possible in that kind of conditions, in that environments. Also, the evidence of prebiotic chemistry. We know that in our planet, life emerged at one point in our past. So we need to know about the exact conditions in that moment for life to arise. So we try to search for these kind of conditions elsewhere, not only in our planet, but outside our planet. And then we need to know about the life itself, the evolution of life. We have a very, very living, evolved planet. So we need to know if there is another places with not so evolved life, or at least with life in the very, very, very early times of life. For doing that, we need to understand life itself, and then we need to understand the evolution of the universe. When is it possible in the universe for life to exist? Not in the beginning, because there is no matter at all. So we need to understand also that. And then we have the help of the techniques of the technology for doing space and planetary exploration. We need to know about the origin of the elements and molecules we are doing by matter. Living beings are made by matter, so we need to understand the creation of matter first. Here we can see that the word chunks means all the chemical elements that are devoted to life: carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. All these elements are on our planet but are elsewhere. So we could think that it could be possible for life to exist elsewhere because these elements are available for life. And that's a very, very important thing, because we could develop some searching techniques to understand environments outside Earth, to ask these environments if it's possible for life to arise. From molecules to life, we need to know also how it's possible for a habitable world, habitable place, habitable environment to develop life in these kind of places. So we need to understand these steps, from prebiotic synthesis, polymers, vesicles, protocells, and cells itself, and then all the diversity of life on our planet. The limits of life are also important. We know our tree of life on Earth. We know the terrestrial biochemistry, but we could ask for another kind of biochemistries outside Earth. And finally, technology for space exploration allow us to understand all these environments by analyzing on site these conditions. So it's very, very important for us, because this is a very, very, very important moment in humankind to understand about life in the solar system and outside. We have planetary explorers and also cosmic observers that are helping us to understand all of this. So I want to show you briefly some experiments that we can do with our students to let them understand, to let them know more about astrobiology, or the techniques, the analysis of environments in astrobiology. First of all is regarding life on Earth, to know about if it is life in a sample. So we can try to analyze a sample and try to understand if it's life present in the sample by extracting DNA. So this is a very, very common process, laboratory process in our schools. So we can do with our students this DNA extraction in order to know the presence of life, of cells in our sample. So we can make this process with students to make them understand about the real science, analysis, how is the process, and the exact measurements and the analysis of the sample to be able to know the presence of life in our sample. You can see here this link, a video in Spanish, but it's very explained and it's very easy to follow. Or we can do a game live to try to understand the DNA replication. This is a very, very important process in life. The DNA replication, the information, the code information in our DNA molecule, it can be replicated. So we can do this live. We can try to explain our students how is it possible for a molecule to replicate itself, and we use enzymes. This is the unzipper one, the initializer, the gluer, and the builder. These are the real names: helicase, polymerase, primase, and ligase. And we can do that live with our students, like a game, by using people instead of nitrogenous bases, and also by doing the hands to replicate bonds, phosphorus bond and hydrogen bond. And we can do this like a game, making the enzymes also people that know how to do it, how to open the string first, how to put together the different pairs of bases, and then create the two streams to replicate the first string, the initial DNA molecule. So this is like a game, and it's very, very, very interesting to do it with people. Here you can see with colored caps to simulate the bases, and then with a string to do the phosphorus string in the DNA, and here you can see the result, the 2 strings. And this is very, very interesting way in which students can feel how the DNA replication is done. It's very simple. We can do also impact craters, simulate impact craters to make students understand about the creation of this kind of features in the rocky surfaces of planets and moons. And we can try to make them understand the study of the composition of the different layers. So we can know more about this kind of environments and the possibility of life to exist in this kind of environment, because we know how to analyze the composition by analyzing these features, these craters. We can try to simulate these real ones by ourselves using flour and small balls, and we can try to replicate these craters to make them understand simulate, to create the model, the explanation model, to try to understand what is going on. Regarding water on Mars, it's very important for life. Water, we know a lot about Mars, and in the landing sites and also in the surface, we discover features regarding the presence of liquid water in the past. So we can ask for why there is no liquid water now on Mars surface, and we can do some easy experiments by using hot water, not boiling water, but hot water, and a syringe. And by opening the syringe, and we can make, as you can see, boil the water, but not because of the temperature, but because of the dropping of the pressure. So we can explain students that on Mars surface now, the lack of atmospheric pressure is the reason why there is no liquid water now on Martian surface. And finally, we can study also other worlds. So we can do activities regarding exoplanets. We know a lot of exoplanets because we have a big amount of space telescope and ground-based telescopes discovering a lot of exoplanets. We know about the new family of exoplanets, super-Earths, Neptune-likes, gas giants, hot Jupiters, X-size, and so on. So we know a lot of them. We have a lot of images of these exoplanets, but these images are fake. There's no real, because there's no information about the composition of the surface, but only the size or the density, but not these colors. So this is fake. So we can imagine exoplanets with the student. We can give them information about the exoplanets and let them imagine these exoplanets and create exoplanets with this data, trying to understand the possibility of life in that places. We can do it by drawings, or also by using these kind of spheres resembling the sizes of the exoplanets, gas giants, or terrestrials, rocky ones. And we can do that with a classroom, and we can put all the planets together, and we can create planetary systems and so on. So it's a very, very interesting way of work with the students. And with these kind of experiments are very easy to replicate. We can make them understand about the astrobiology and make them understand about the kind of research that astrobiologists are doing. And that's all from my part. Thank you very much. And I hope you enjoy this and you use them in your classroom. Thank you very much.